fermentation, what is the point? Do you remember carbohydrate fermentation?
Go on, Dr.
Beautiful.
Russell.
Dr. Vazentin's nailing it over there. If if it ferments carbohydrates, ferments. You get the point. It ferments carbohydrates. What were our 3 carbohydrates? Lactose, sucrose,
Glucose?
glucose. So we had lactose, sucrose, glucose. What else did we have in the tube?
Oh, was that the one we did citrate?
It was a little bit.
You don't remember what it's called? So it's a Durham tube. Durham.
Oh, the little tube inside the tube.
Durham? I'm not sure if that's how you spell it. It's a Durham tube. So remember, it's upside down inside the carbohydrate.
And if it ferments, then there will be gas bubbles or
Yes.
displacement present in the Durham tube.
Yes. So we're looking for gas. What kind of gas is it?
Carbon dioxide?
Yes. Beautiful. What does A stand for? Acid. What? So pH, lower pH. So that's phenol red. Like Dr. Fuller said, phenol red is the same. It's the same pH indicator. Lower pH acid means it is a fermenter. So automatically we know if it ferments, then it's facultative. If it doesn't ferment, we don't really know. Okay, gelatin.
Could you just write that down, facultative?
Facultative.
Thanks.
I was getting fancy with the colors earlier. Now I'm going for green.
All good.
Okay, gelatin. What enzyme are we looking for? Or let's talk about what the whole point is of gelatin. So remember what it looked like after we let it set and after it went in the fridge? So gelatin,
Ours didn't.
what is the enzyme? Gelatinase.
Gelatinase.
Which is a what? Do you guys know what gelatinase is? Do you remember broadly?
An enzyme?
It's an enzyme. It's also an exotoxin. So this is also an exotoxin. Because we are made of gelatin. We have all of our connective tissue is made of gelatin. So if you have a gelatinase enzyme, that's not good. An organism that spits out toxins that degrade connective tissue. That's not good. So it's an exotoxin. It's a gelatinase enzyme. Remember, gelatin is, what was funky about this lab? What did we have to do?
We had to refrigerate it.
Refrigerate it.
Inoculation.
So
Or incubation.
gelatin at room temperature, or at what temperature is that one?
Is that the 37?
37. 37 is what temperature? 37C is what temperature?
Is that
What did we say, Dr. Long? Do you know? Yes. Beautiful. Human body temperature. Awesome. So it's about 37 or 98.6. Okay, so room temperature or at warm, it's well, I don't want to say room temperature. Let's say when it's 37 degrees, it will be liquid. After the fridge, it's solid. But if it's positive, then even after in the fridge, it's still liquid. Even after it's been refrigerated. Yeah? Because it chewed up all the gelatin, so now it's just liquid. Got it? Okay, oxidase, what are we looking for in the oxidase? What is, remember when I said oxidase, you always say the blank oxidase. Sorry, it's like a lot of little ones. What is that? There's 2 words that go with oxidase that reminds us what the oxidase test is looking for. Close.
cytochrome?
Remember it's a two-part word, blank oxidase.
Cytochrome.
Cytochrome. Cytochrome oxidase. So cytochrome oxidase, so you're looking for cytochromes. So remember on that inner membrane of the bacteria, remember we're still in prokaryotes, you have those cytochromes all the way along. So we're looking for one of these cytochromes. So cytochrome oxidase test. So if it's cytochrome oxidase positive, what does it look like? What is the test? What is it?
Does it bubble?
What is it?
Does it?
We did these two together. This one, students always get confused between these two because we do them together.
Oh wait, is that the one that turns purple?
Yes.
Oh.
Yes. So it's the strip that will change colors in the first 15 seconds if it turns purple. We'll put Sharpie on
Strip.
it.
Yeah, I have to go through all this at the beginning anyway. Oxidase
So if
positive
it
turns
turns, if the
blue
little
and gets
strip
progressively
turns,
darker.
then it's positive. And that means it has the cytochrome. And what does that tell us about the organism? Yes, way to go, good. Aerobic. Aerobic. Or facultative. Don't forget that. It doesn't mean it's automatically aerobic, it can also be facultative. Okay, catalase. What is the whole point of catalase? Let's just talk about what the catalase test is. What are we doing on the test?
It differentiates staph bacteria.
Yeah, we're definitely looking for differentiation. What is the test?
You're looking to see if they produce that enzyme.
That's
Yes, if they have the catalase enzyme, awesome. Yes, so they have the catalase enzyme. So what are we putting on here? Hydrogen peroxide. So if we're putting hydrogen peroxide on here, and if we put hydrogen peroxide and the organism on here, then it does, what happens?
Bubbles.
If it bubbles, it's positive. So if it bubbles, it's positive, that means it has the catalase enzyme. So then the bubbles are going to come out, so it's going to go H2O2 to O2
Plus H2O.
Then what does that tell us about this organism?
Is it?
So remember oxygen down here of the cytochromes. Remember oxygen is the final electron acceptor. And so oxygen plus the hydrogens usually make water. So the hydrogens that come down the ATP synthase, the hydrogen will come together with the oxygen and make water. But sometimes it makes a mistake and it'll make hydrogen peroxide, which is toxic. So our bodies, just like a lot of facultative organisms or aerobic organisms, will have this catalase enzyme so that we don't have this toxic H2O2. It's kind of like a checkpoint system to make sure that we don't have hydrogen peroxide running around. Okay, dichotomous key, that's easy. You guys will